webinar sponsored by Swami Vivekananda Research Center. Ramakrishna Mission Vidyamandira began its journey in 1941 following Swamiji's vision and comprehensive scheme of education. The college was upgraded to a 3-year degree college in 1960. Till now, this college has achieved many milestones. In 2013, the college was reaccredited by NAC with grade A. In 2014, the college was awarded the status of college with potential for excellence for second time by the UGC. In 2020, our college ranked 7th in the India and first in the West Bengal in NIRF ranking. Department of Zoology is the youngest department of Ramakrishna Mission Vidyamandira, established in 2015. From 2017, our department have started 3-year degree course in zoology honors. With this very short period of time, our first honors batch have come with colors. Now, I would like to request our principal, Swami Ekachittanandaji Maharaj, to deliver the welcome address.
Om Namo Bhagavate Ramakrishna. This guest who might be attending this webinar, respected Vice Principal Maharaj, Professor Avijit Dey and his associates and beloved students. My heartfelt namaskar, respect and good wishes in due places. Especially on behalf of Vidyamandira family and the Department of Zoology as well, I extend a most hearty welcome to Professor Sujay Ghosh. Due to this existing pandemic situation, we are not being able to conduct the seminar in a real platform, but thanks to the scientific advancement, we are getting connected. So our head of the department has already given a short introduction to the institution's vision and mission. And to sum it up, actually this is the outcome of Swamiji's dream of Swami Vivekananda, that the Ramakrishna Mission should strive hard to combine Eastern prudence with Western science and technology. And I'm very happy that though the Department of Zoology, as Professor Dey mentioned, is the youngest department, it is full of enthusiasm, full of resources, and they are trying to combine Eastern ethics with Western technology. And there's a good news as well, which I would like to share with all my beloved students and the members of the teaching faculty of the Department of Zoology, that our Professor Uttam Kumar Ghodoi, the head of the Department of Industrial Chemistry and Applied Chemistry, and his associates have come up with flying colors, and their one publication has come recently, been accepted in American Chemical Society Nano. The news has come today only. So we are very happy that on this gala day, we are organizing this webinar. I'm confident that the enlightening discourse from Professor Sujay Ghosh will be much enriching experience to both the students and teachers alike. Once again, I heartily welcome Professor Sujay Ghosh and hope that in the near future we can meet together soon, very soon. Om Shanti 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 Hari Om Tat Sat Shri Ramakrishna Arpanamastu.
Your valuable speech. Now I'd like to request our faculty member, Mr. Krishnendu Ghosh, to say a few words.
Thank you, Eddie sir. Very good morning to all of you, and especially the delegates as well as the respected teachers, and most of all Dr. Sujay Ghosh, our very own Sujay sir, from the Department of Zoology, and we really miss you, sir, in these days. So, before I say anything, I can just remember one thing in my childhood that I once asked my elder sister during, I was in class 3 or 4, I guess. And at that time, I asked her, uh, what is cell? In Bengali, কোষ ব্যাপারটা কি? What is cell? And she told me that it's everything. It's everything in life, and it is the factor by which you are standing here. And after that, she went on and said that from this day, you just remember one thing, that there should be a Pandit Moshai, and that Pandit Moshai should have an Astotiki. That means prophase, metaphase, anaphase, and telophase. Pandit Moshai Astotiki. That was a very fun thing, and with that frolic, I came to know about the basics of biology. And as the time passed on, I just realized something, that in mathematics, what seems to be very, very easy thing, that means 1 plus 1 is equal to 2, seems to be most difficult type of thing in case of biology. Because when we mix n plus n equals to 2n, I think it is the most intricate thing that a biologist can ponder upon. I think so. And for the as the time passed on, I actually used to ponder upon one thing. Why Didi told me Asto? What is the significance of Asto? Why should I tell the Astotiki? And later, I came to know the significance of anaphase. And if there is no anaphase, I think the entire cell cycle and the cell will be at no use. But in this case of anaphase, we know that a mild and most insignificant type of thing can create an havoc, a term known as the non-disjunction. A simple thing that could create havoc is the responsible factor for a syndrome known as the Down syndrome, in which Sir is going to tell us with all his expertise. And for that, I am very much honored to be a part of this webinar, because from 2020, due to this pandemic, it is very difficult to have this kind of talk in real platform, but due to the virtue of this Google Meet or something, the online platforms, we are very much lucky to have this kind of webinar that will enrich us, all the students and the teaching faculties, with some of the gracious knowledge and research outcome by our Sujay sir. So I think we will all are very much eager to listen to your valuable talk, sir. So with this, I am very much sure that the day will be a good kickoff with your speech. Thank you, sir.
Thank you, Kuji sir, for your beautiful speech. Now, we are really fortunate to have Dr. Sujay Ghosh as speaker of this webinar. Sir has completed his MSc in zoology from University of Calcutta in 1999. He has completed PhD from West Bengal University of Technology, Kolkata in 2009. He is currently appointed as Senior Assistant Professor in Department of Zoology, University of Calcutta. He has successfully completed many research projects funded by UGC, DBT, ICMR, WBDST. His research area is mainly in human genetics and Drosophila genetics. His research is mainly focused on identification of genetic, epigenetic, and habitual risk factors of trisomy 21 and chromosomal non-disjunction, genetic and epigenetic risk factors of male infertility, effects of common toxicants on the genetic regulation of developmentally relevant genes, in imaginal disc in Drosophila, evolution of immune response among the related species, and local demise of Drosophila. Sir has received many national and international awards. Till date, Sir has guided 2 PhD students and as principal investigator. Currently, 4 research fellow is working under his supervision. Sir has already published many book chapters and 45 research articles in renowned international journals. Sir has discovered and reported 61 new mutations, including transition, deletion, frameshift mutations in relation to chromosomal non-disjunction and male infertility from Indian Bengali population. With these few words, I like to conclude and request Dr. Sujay Ghosh to deliver his talk. Over to you, sir.
Thank you. Am I audible?
Yes, sir, you are audible.
And the screen and slide is visible?
Yes, sir. Yes, sir.
Okay. Okay. Thank you. Thank you very much for these nice introductions. And at the beginning, I should convey my heartiest indebtedness and gratefulness to the Maharaj, Principal Maharaj, as well as Head of the Department of Zoology, and all other faculty members of the Department of Zoology, Ramakrishna Mission, for giving me this opportunity to deliver this talk and share my thoughts and idea on science as well as in society, particularly in the field of Down syndrome or trisomy 21, because you, the youngster, are the future scientists. So this is our responsibility to ignite your mind with some new thoughts and ideas so that we can hand over our torch for carrying this responsibility of the research and its application in the society to the next generation. And as I know that students of Ramakrishna missions, they have very bright, intelligent mind, so that I think this today's lecture will be one of the favorites of them for their future work, and they will be inspired by this talk. Whatever, but what I want to say that if you are ready to hear, I can speak in Bengali, because this is our vernacular, our mother language, and what I feel that Bengali as a language for interactions appeal our mind more incisively than any other language. Well, English will come as natural way, but if you agree, then I can proceed with Bengali so that you could catch some tidbits and beats and some complicated part of this lecture very easily. So,
sir, I have one request.
Yeah.
I have few students from different parts of this country who are not at all conversant.
Okay.
So, like that.
Okay.
So you can mix it.
Okay. Sure.
Yeah.
So at the very beginning, I should extend my respect to Thakur Sri Ramakrishna, Sri Ma, and Swamiji for enlightening us throughout our life. So just with these few words, I'm going detail into my lecture. So my title of the lecture is Down Syndrome, Risk and Recent Crisis. You can see this backdrop of this title slides is actually our Calcutta University Darbhanga building. This is a great historical building. So there is some facts about Down syndrome. This is most common live-born trisomy in human and is caused by trisomy 21. That means the triplication of chromosome 21. The global incidence of trisomy 21 is 1 in 700 live birth, and it is actually the leading cause of mental retardation, all recognized medically identified mental retardation. And trisomy is also a leading cause of pregnancy loss. That means whatever the number of child born with the trisomy 21, nearly about equal number of fetus is actually being aborted during any time of the pregnancy. So at the same time, it is one of the major cause of pregnancy loss, as well as at the same time, it's a leading cause of the mental retardation. So this is a great man in 1866. This is John Langdon Down, who first wrote a paper entitled Observation of the Ethnic Classification of Idiots. And this is a very pioneer and initial paper that described the Down syndrome first. Nearly after 100 years, in 1959, this great man, this is Jerome Lejeune, identified this particular set of idiots, because this term idiots was initially used by this group of medical practitioners or physicians to describe trisomy, and later they used the term Mongolisms for having the similarity in the appearance with the Mongoloid peoples with this trisomy individual. And this Jerome Lejeune, the great man in this field, identified the cause of this mental retardation is presence of extra copy of chromosome 21, and he described the condition as trisomy 21. This is a karyotype that shows the presence of extra copy of chromosome 21. Students, you can see this karyotype. You are familiar with this karyotype when all other chromosome sets are in the diploid condition, but chromosome 21 has triplication. This excess amount of the genetic material create a lot of problems, but we are not going into that, but rather our focus area is what are the causes that actually lead to presence of excess number of chromosome in the oocyte or in the gamete. So what should happen in the ideal condition? Again, very familiar diagram. In the ideal condition of the cell division, there is equal distribution of 2 homolog chromosomes in meiosis 1, followed by separations of sister chromatid equally and perfectly into the gamete that lead to the formation of the haploid chromosome set in each ideally healthy gamete. But in case of the non-disjunction, there is some reasons for which chromosomes do not segregate, or if the chromosomes segregate properly at meiosis 1, but sister chromatids do not segregate properly at meiosis 2, and that lead to the production of the 2 different types of abnormal gamete. One has extra copy of chromosome and another have no chromosome. So the gamete having this extra copy of chromosome may receive another copy from the opposite side, that is the opposite sex, during the fertilization, and that lead to the formation of the triplicated conditions of particular chromosome set. In case of the normal meiosis, 2 homolog chromosomes, they come close together at the pachitin of the meiosis 1, at the pachitin of the meiosis 1, and they recombine with each other, and the recombination takes place ideally at the middle of the chromosome arm. That recombination has very important implication in chromosome segregation, and that is very usual. All the chromosome pair within the oocyte experience that kind of single recombination or double recombination, and that recombination event actually maintain the pull from the opposite pole, and ultimately the balance segregate properly, and even the sister chromatid gets segregated in the oocyte. But in case of the meiosis 1, due to some the reasons, age of conception, or if the women like to have baby in their advanced age means beyond the age of 32, there is an increased chance of having that kind of the baby with abnormal chromosome number. So this is the first, and after having some signal from some test, you may interpret or anticipate that fetus growing inside the mother's womb may have certain problem. So one is very, very common ultrasound. We usually do ultrasound for all kind of the pregnancy, but the ultrasound results may have some indications in case of Down syndrome baby, and careful observations may lead to identification of trisomy 21 conditions in this fetus. And this is some figure of the Down syndrome fetus in ultrasound, and that region you can see, this is called nachal region, and this nachal region exhibit almost transparent area, which is very unlikely in case of the normal healthy baby. This condition is called nachal translucency. This is one of the criteria through which we can interpret the developing fetus may not have healthy developmental regime. It may have some problem, maybe Down syndrome, or maybe any kind of neuronal disorder. There are some recommended blood tests by which you can monitor the fetal fat even at the time of pregnancy. One is maternal serum alpha fetoprotein, which actually goes down if there is Down syndrome baby than the normal baby. Estriol, one of the female hormone, particularly in case of pregnancy, there is an excess amount of the estriol in the maternal serum. But if the Down syndrome baby grows in fetus, the estriol level goes down than the normal level. And human chorionic gonadotropin, that hormone usually present in the optimum conditions, optimum level in the maternal serum, but it becomes elevated when there is a Down syndrome pregnancy. Inhibin A, another important hormone from the gonads, that also exhibit increased secretion in case of the Down syndrome pregnancy. And PAP-A, one of the important protein marker in the maternal serum, that goes down in case of the Down syndrome pregnancy. Collectively, these are known as pentaset testing for the Down syndrome, and from this testing you can interpret the baby is going to have extra copy of chromosomes, and these indications lead to the another important test known as amniocentesis. Amniocentesis is nothing but to collect the little amount of the amniotic fluid from the maternal womb and look into the cell, that is WBC, which is coming from the fetus, and count the chromosome number. It is actually the classically karyotyping, but the tissue is taken from the amniotic fluid. So it is known as amniocentesis. Apart from that, in the very recent time, we have PCR technique and we have several DNA marker, and using short tandem repeat marker and PCR, you can go for the molecular testing to count the chromosome number. And it is very simple. If you look at the gel diagram on the right side, you can see in the middle of this gel electrophoresis lane, there is three distinct band. These three distinct band actually count of the three distinct chromosomes genotyped using STR marker. And very importantly, the advantage of this technique is that you can monitor the origin of the extra copy of chromosomes either from the mother or either from the father. If you look at this diagram very carefully, you can monitor that the single band of this father can matching with this middle band of the Down syndrome baby, and two bands, that means two chromosomes of the mother, are absolutely similar in the position with the two bands of the Down syndrome. So the extra copy of chromosome is actually maternal in origin in case of this example. Well, here too on the left side photographs, you can see there is a thickening of the band. And this thickening of the band means two bands remain clamped there, and they are actually the two band, that is double chromosome. They are representing the double chromosome count, and this double chromosome is coming from the maternal single chromosome. That means during the course of this inheritance, the maternal single chromosome get duplicated, and then duplicated copy of chromosome enter into the oocyte, lead to the Down syndrome childbirth. So in that way, the advantage of the DNA typing of the parents and fetus is that you can identify the origin of extra chromosome unambiguously. This is something advantageous over the classical karyotyping. So we started research with women having Down syndrome baby to look into the risk factor, and the key question of our research were how does recombination affect chromosome segregation? and how recombination frequency and pattern interact with maternal age of conception. That are not for generating the variation in the genome, but also to ensure the proper chromosome segregation during the meiosis. So there was an initial publication by a group of scientists who happen to be my colleagues right now, my collaborator from USA. They identified and published the paper in Science that recombination may have some implication in chromosome segregation. And we started working with this hypothesis to identify how recombination affects the chromosome segregation. And importantly, another risk factor that was pointed out by the Penrose in 1933, that maternal age, if recombination is one of the risk factor and maternal age is another risk factor, then how these two risk factors interact with each other, that was our another query. So we started with this hypothesis and we start genotyping the entire chromosome 21 with sets of these microsatellite markers. We segregated the chromosome, entire length of the chromosome 21 in 6 intervals, and from each interval we genotype these short tandem repeats, that is STR marker, to identify the recombination profile. And again, each marker can generate that type of photographs, and from these photographs we interpret whether the chromosome is exactly inherited from the mother or chromosome get duplicated during the inheritance. If it get duplicated during the inheritance, we will identify the state of this marker as reduced state, or if it is inherited just as it is in the mother, then we identify it as non-reduced marker. In way, all 50 markers that we use to genotype the chromosome 21 from mother as well as from the baby come at least that kind of the string generation, that is information string, where the individual marker may be non-reduced or may be reduced. If there is a break from non-reduced to reduced, then we will interpret a single event of recombination. This interpretation is little bit complicated, and it's really complicated to understand at this lecture right now. It is something statistical, and it based on the Bayesian statistics of probability. So using statistical knowledge, our collaborator friends who is statistician as well as geneticist actually designed this program to look into what is actually the status of the recombination can be interpreted in this genotyping data. So I am just going to show you, this is our family data string. So individual Down syndrome family, that is PS1, PS2, PS3, PS4, all these are Down syndrome family. An individual horizontal line is actually presenting a single chromosome 21 from each mother. And you can monitor, there are N. That means these markers are actually showing the non-reduced status when it comes to the baby, and R is actually, you can look at this R. So up to that point there is N, that means all the markers, they are non-reduced, and then there is an R, that marker is getting reduced, and whenever there is a break, that means N to R or R to N conversions, we will interpret it as a single recombination event at that point. In that way, we went on creating this data sheet for thousands of the family to look at you what is actually happening in the population level. And this is software known as NDJ map, and it is designed on the hypothesis from expectation maximization algorithm of Bayesian statistics. And our friend and collaborator, Professor Eleanor Fingold from the University of Pittsburgh, designed this software where we can put this data of genotyping, and software will show you the recombination value. These are the output of the software, where the lower strata actually showing, it's present in the red color, actually showing the exact recombination value in individual intervals. So in that way, we came to conclude and estimated that maternal non-disjunction error constituted about 92% of all incidents of the Down syndrome. Paternal error contributed very negligible, that is 8%. Within the maternal error category, meiosis 1 is the major cause of non-disjunction or stage of non-disjunction, whatever you like to say. It representing about 70% of the total incidence of all maternal error in the oocyte, and meiosis 2 represents 27% of the error in the oocyte, and only small fraction, nearly 3%, is actually the post-zygotic error. In case of few paternal non-disjunction error, we were able to again segregate and identify them as meiotic 1 error or meiosis 2 error, and it's about almost equal 42% and 54%. So we focused our further study onto the maternal meiosis, particularly on the meiosis 1 error, which constitute the bulk of the cause of Down syndrome baby in our society. So we started creating some model where, again, these are some statistical model where we can show as well as test the interaction between maternal age as well as recombination. We segregated all the participating mother into the 3 group: young group, those who have Down syndrome baby below their age of 29, we called them as a younger group. Then middle age group actually constituted with the women who have a Down syndrome baby between their age of conception 29 to 34, and the older age group who had their Down syndrome baby beyond the age of 34. Very surprisingly, you can see that whenever Penrose told that maternal age is one of the important factors, that means advanced maternal age, there is an increased rate of Down syndrome baby, but we had our sample having Down syndrome at younger age too. I'm coming to this point later on. And then we distributed all these meiosis event. Meiosis event means recombination event. That is, 1 means single recombination event, 0 means no recombination event, and 2 means more than single recombination event as a function of the maternal age, both in meiosis 1 cases and meiosis 2 cases, and go for further statistical analysis. And again, 2, we placed all the recombination event into the 6 interval as a function of maternal age, both in meiosis 1 and meiosis 2 category, and got that kind of the results, which is actually interpreted in that way. I am coming to this photograph because this is actually understandable to you. We found 3 risk factors in summary. 1, there is no recombination. That means ideally recombination should be there at the middle of the chromosome arm to ensure proper chromosome segregation. But we found that among the younger mother, particularly in the meiosis 1 error group, there is no recombination. So we can conclude that no recombination, that is absence of recombination, is a great risk factor for non-disjunction at meiosis 1 of oocyte development, particularly in the younger mother age. Secondly, we identified one very close telomeric exchanges. These telomeric recombination was found frequent in meiosis 2 group. That means women who had error in the meiosis 2 often exhibit telomeric recombination of chromosome 21 at their older age. And third one, we found there is an excess amount of the centromeric recombination very close to centromere. So these 3 recombination configuration were identified through the statistical modeling that no recombination, telomeric recombination, and centromeric recombination, these are 3 important risk patterns that may cause non-disjunction and may cause non-segregation of the chromosome properly during oocyte development. So we published paper in this American Journal of Medical Genetics in early 2009. And our next venture is to look into environmental and habitual risk factors that are very prevalent in our country, because our mothers are exposed to different kind of pollutants and may have some risky habits like tobacco use in our society. Cigarette smoking is very risky for healthy oocyte development, but these are not very prevalent in our society. But we found through our epidemiological study that in our society, particularly in a particular class, the females are exposed to chewing tobacco. So we found two important factors. One is chewing tobacco, as I told you, and another one is irregular contraceptive pill use. These are dangerous practices by today's girl, because they just go on taking medicine just by purchasing from the medicine shop without taking permissions or without taking consultations from the doctor. So we again model different statistical pattern and found there are some interactions of these risky factor with the genetic factor, and these are some of the statistical modeling. I am not going detail into that because these are some are very tedious and maybe boring to you. I'm just coming to the conclusions. We published this data in the American Journal of Epidemiology from the Oxford, and where we show that smokeless chewing tobacco is a great risk factor for absence of recombination. That means the girl who started taking smokeless chewing tobacco in form of gutka or in any other form from the early days of their childhood, in some part of the society, they start taking this chewing tobacco very early at the age of 7, 8, 9, or 10. They gradually acquire some increased risk factor of having that kind of the problem, because chewing tobacco in any way disrupt the chiasma formations or some affect some of the protein component of this chromosome segregation system. So that led to non-disjunction to a great extent. Now, our third venture is to look into how does molecular aging associated with chromosome 21 non-disjunction. The hypothesis was that, as I have told you, that some of the women from our society, they have Down syndrome baby even at their age of 20, 21, 22, 25. That means so-called young age. But still, instead of their young age, they have high risk of the Down syndrome baby. So suddenly it came in our mind whether these group of ladies suffer from molecular aging or not. That means if you, for example, have two group of lady, one have 20 years, average 20 years age of conception, another have average 20 years age of conceptions, but one have rapid deteriorations of their molecular apparatus in the oocyte, we may call this event as an accelerated molecular aging, but another doesn't have that risk. So we wanted to discriminate these two silent molecular aging process in these two groups. So we started looking into the telomere. As you know that telomere is a good signature of molecular aging. If there is a molecular aging, there is rapid telomere attrition. If there is no molecular aging, there is slow telomere attrition. This is established hypothesis right now. So keeping this fact in mind, we started looking into the telomere length of these two group of mother. The mother having Down syndrome child have their very early age, that means 20 to 25 or 26. And the mother, they have no Down syndrome baby, but have the same age. And obviously the older mother that have Down syndrome baby in their older age. Very surprisingly, we found that you can look at these gel photographs. We found that the control mother have longer telomere. As a students of molecular biology, you know that if the size of DNA is bigger, they return in the gel matrix. So they move slowly through the gel matrix and remain at the upper part of the gel. This is very usual. But if the DNA fragment is smaller, the smaller fragment will migrate faster and they will come at the bottom of the gel. So easily you can compare these two groups just by looking at this photograph. They have larger DNA fragments and they have smaller DNA fragments. We hypothesized that that particular group of mother, those who have Down syndrome child at their very early age, may have some accelerating aging, molecular aging, and that molecular aging is reflected into very rapid attrition or shortening of telomere length, whereas other mother, those who have Down syndrome maybe at the latter part of their life, that is at the advanced age, beyond the age of 32, they have usual pattern of telomere attrition. So that was one great hypothesis. We go for statistical modeling to estimate what is the rate of attrition of telomere length in the control mother. We found that it is only 61 base pair. That means the mother who have healthy euploid baby, they have the telomere shortening length only 61 base pair per year, whereas in case of the mother who experience meiosis 1 non-disjunction, that is M1 non-disjunction, they have accelerated rate 110 base pair per year, and in case of the meiosis 2, the rate of telomere attrition is much higher, 220 base pair per year. So that was something great, and we was the first to hypothesize, and it was very novel concept, and we end up with this publications in the Human Genetics Journal in 2010. It's very prestigious journal, and telomere length is associated with type of chromosome 21 non-disjunction, new insight into the maternal age effect on Down syndrome birth. Later on, our collaborator from the US said, that is specifically the Stephanie's group, I will show you the picture of the Stephanie at the end of my lecture. They got a lot of interest into looking into it. That was the very initial publications, and they took nearly 5 years to replicate the entire study on the American population, US population, and they end up with the same conclusion in the same journal. They also published the same thing in the journal Human Genetics in 2015, replicating our entire work on Indian population, and they found that telomere length is somehow associated with chromosome 21 non-disjunction, and somehow telomere length attrition may impose some rapid molecular aging that is not discernible from the outside. That means you could not identify this young lady actually have the rapid molecular aging inside, but she's at the higher stake of having Down syndrome baby when she will be pregnant. So these are some discrimination with incisive study, combining molecular study with statistical tool. So our hypothesis was the mothers of Down syndrome are genetically older than the mothers of the normal euploid children, particularly at their older chronological age. And in some way, the molecular apparatus of telomere maintenance is linked with molecular apparatus of chromosome segregation system and pattern of non-disjunction. In the next phase, we started looking into whether this habitual risk factor, that is tobacco use, have some effect on the telomere shortening or not, because in our epidemiological data set, we saw that the mother who were suffered from the telomere shortening have, in maximum cases, have the practice of chewing tobacco taking from the early of their childhood. So we again go for some incisive molecular and statistical modeling, and from these all statistical modeling and analysis, these are really complicated. I'm just skipping these slides. Analysis, we found that smokeless chewing tobacco and shorter telomere length, they are associated among the women who experience meiosis 1 non-disjunction. That means, in other way, we can say these hypothesis in another sentence, that smokeless chewing tobacco may accelerate the telomere shortening rate, and as the telomere gets shorter, it increases the meiosis 1 non-disjunction. Secondly, smokeless chewing tobacco also exhibit some association with meiosis II non-disjunction, particularly at the older age. Because in case of the older women, there is an natural deteriorations or declinations of protein machinery in their oocyte. So in addition, if there is chewing tobacco, that will create some more complicated situation, and that led to meiosis II non-disjunction. That means chewing tobacco is risk factor both for meiosis I non-disjunction at the young age and meiosis II non-disjunction at the old age. So we published this paper again in a very prestigious journal, Public Health Genomics, in the year, it's not very old, in the year 2015. Then we started looking into the candidate gene polymorphism. So what we did from initial publication of 2009, we have identified recombination pattern. We have identified that mother have rapid molecular aging. We have identified that smokeless chewing tobacco has some environmental impact on the molecular aging that led to abnormal chromosome segregation. In the next venture, we started looking into the candidate gene. Our hypothesis was probably there are some mutation and polymorphism in the gene that regulate the chromosome segregation on one hand and regulate recombination in another hand. As I told you, that recombination anomaly is the cause of meiosis I non-disjunction. So it is an intuitive hypothesis. The genes which regulate recombination, if have some mutations in their structure that may perturb their normal functioning, that may lead to abnormal recombination, and which in turn associate the meiosis non-disjunction. So initially, we took and identified the SPRDM9. It's very important. You can go for this gene also in the web resources like NCBI or Genome Data Bank. This PRDM9 is a very important gene for all types of primates for regulating their recombination. This is a conserved gene in the primate evolution. In all apes, chimpanzee, gorilla, all they have PRDM9, including human. So we started scanning the PRDM9, whether our PRDM9 carries some mutations among the women that may cause abnormal chromosome segregations and abnormal recombination. Yes, we got and identified certain mutations through the gene sequencing. We found there are certain mutations in the PRDM9 that may perturb the normal functioning of the PRDM9 during the course of meiosis recombinations, and that led to no recombination at all. As we saw in the earlier slides, that no recombination, that is achiasmate meiosis, we often call this achiasmate meiosis or no recombination meiosis maybe have a background of these PRDM9 mutations that led to non-disjunction. Then we looked into the second gene, that is presenilin-1. Presenilin-1 is a protein component equally expressed in brain as well as our gametes. In gametes, presenilin-1 remain associated with centromere, and they ensure proper segregation of sister chromatid, particularly during the anaphase. So again, we look into the presenilin-1 and we found that this T is actually a mutant. There should be A, but it converts to the T. And there is another one. There should be G that convert to T, that converted to A. So these are mutations that destroy the function of presenilin-1 in the optimum conditions and cause sister chromatid non-segregation. Then we looked into another set of the genes, those who are responsible for regulating folic acid metabolism. And this is MTR. Again, we found that folic acid is very important. As you know that all the doctors and physicians, they do recommend folic acid during the course of pregnancy because folic acid is a good stimulator for the neuronal development, brain development. But we found that not only the neurons of the baby, developing baby, folic acid is important ingredient for healthy oocyte formation. How it works? Folic acid actually ensure proper chromosome segregation and helps in maintaining absolute cytoplasm to nucleus ratio or volume in the oocyte. Replaced by G in this MTR gene in the mother of the Down syndrome baby, but not in the control. You can see this peak. A is converted to G. That destroyed the natural function of this folic acid regulator. As a result, the oocyte could not metabolize the folic acid properly. As it cannot metabolize the folic acid, it commit non-disjunction. And another mutation in the same gene. So this is a control mother who doesn't have any mutations in this gene. So in hypothesis, we may say that chromosome 21 non-disjunction is a multifactorial trait. There are several risk factors which are working at the different phase of the oocyte formation. If we depict this all risk factors into this diagram, we can say during the early phase of the oocyte formation, there is a risk factor of the susceptible chiasma formation. That means I have shown you the telomere chiasma as well as centromere chiasma. These are very risky for chromosome 21 non-disjunction. Secondly, when meiosis 1 arrested naturally, there is a chance of accumulation of the environmental insults into the oocyte. That means environmental factors are there. I have taken only single one, that is chewing tobacco. We may consider these as an habitual factor as well as environmental factor. Then as the woman ages and the follicle grows as a Graafian follicle, there are some chance of degradation of meiotic protein. That means when the woman ages, there is a rapid deterioration that is age-related and normal, this meiotic protein and segregation apparatus. So if you put some environmental insults here, that will accelerate this degradation process and increase the chance of anomalous chromosome segregation. So these are some of the baby. And now come to other epidemiological risk factors. Well, one is obviously that we have identified, even in the Western population, we have identified that smoking is important risk factor. In case of us, it is chewing tobacco. Maternal habit of alcohol consumption is another important risk factor and very much prevalent in the Western society. Exposure to the different pollutants and obviously irregular contraceptive pill use. All these factors destroy the chromosome segregation system in oocyte. So I'm showing you some of the practices in our country. You know that this very rural practice of this cooking generates huge amount of the carbon monoxide, and this lady probably from the early of her childhood entered the kitchen and get exposed to this carbon monoxide, which is a strong pollutant, as you know. And I spent some of the time of my life in studying this Down syndrome in the Sundarban area, and I saw this is very usual practice for the lady to get exposed into the water throughout the day for collecting the seeds of prawn and shrimps and other fishes. And during the course of their exposure in the water, they came in contact with oil spilling from the so-called bhutbhuti, that is small water launch that are usual for the Sundarban area. And these oil spills gradually absorbs in their skin, and that may increase the risk. Obviously, you can see that the child is sleeping on the lap, but the grandmother having the practice of smoking. These are dangerous, alcohol, and contraceptive pills. So up to this part, we are talking about the maternal risk factor. That is the risk of Down syndrome part. Now I am coming to the second part of my lecture, that is the recent crisis. All of you are familiar with this diagram, as you know that COVID-19 outbreak have changed the entire world absolutely in a different way. So Down syndrome children are at the stake of greater risk and mortality. Why? Well, if you are looking at this diagram, I'm just going to show you, there is a huge response of particular mRNA expression, that is IFNAR1. That particular mRNA is a signal for the immune system. This immune system signal get elevated in case of the Down syndrome, but not in the control. All blue boxes representing the Down syndrome and all the gray boxes represent the control. If you look at this diagram, you can see, well, all the marker or the so-called mRNA, which are very important regulator of our immune system, get elevated in case of the Down syndrome, but not in case of the control. This situation a little bit paradoxical, because you may ask me, sir, if there is an elevation of the immune responsive mRNA, that means overactivation of the immune system. No, sometimes excess doesn't mean good. Sometimes excess does means bad. Here is actually the phenomenon. Even if you look at this diagram, these are all CRP, cross-reactive protein, get elevated in case of the Down syndrome, not in control. IL-2, interleukin-2, always exhibiting high rate of secretions from the WBC in case of the Down syndrome children, but not in the control. And IL-10 also get elevated. Why? There is a complicated genetics behind this. If you look at this trisomy individual, as you know, have 3 copies of chromosome 21. There is a gene on the chromosome 21 that is known as interleukin 10. So there is one excess copy of IL-10 gene on the chromosome 21. That elevated level of IL-10 leads to stimulation of other IL from the WBC to secrete more and more. And entire interleukin family becomes elevated in their expression in case of Down syndrome child. This phenomenon is called cytokine storm. These cytokine storm have immunosuppressive effect. That means this excess amount of cytokine immediately bind with their respective receptor on the WBC and suppresses the natural response of the WBC towards antigen. So trisomy 21 people, they are at high risk of not only the SARS-CoV infection, but any other kind of natural flu and respiratory infection. Rather, Down syndrome children, they suffer from the respiratory distressness, which is not present in the euploid normal one individual. So this cytokine storm make the Down syndrome more susceptible. So you can hear this IL-10 and so many other 3 copies of those who are regulator of the immune system in the trisomy conditions. There is IFNR overexpressions. That INR overexpression lead to hypersensitivity. So increased cytokine productions by the immune cells and increased risk of the downstream pathology. Downstream pathology means here it is the suppression of the WBC response to all kind of cellular and humoral immune response against the any antigen. So we started working immediately following this outbreak in the March 2020. Our Trisomy Research Society Netherlands, that is the international body with which we all, those who are working on the Down syndrome area, are linked. They constituted one global task force. And this global task force principally participated by scientists from the US and UK, and fortunately and unfortunately me only from the Asia. So we started taking sample from the different Down syndrome family through questionnaire makings and epidemiological study. And this first slides is the outcome of this global study, where we are just trying to show what are the common symptoms of COVID among the Down syndrome children as a function of their proportions. So fever seems to be present in all kinds of the children, and followed by cough, then shortness of breath, then there is nasal signs, means some problems and disturbance, and runny nose in the children, then short throats, headaches, and a significant proportion of the Down syndrome children, they exhibited diarrhea. You know that diarrhea is in terms of COVID infection. In many cases, the patient admitted to hospital with the symptoms of only diarrhea, no fever, even for the euploid normal population too. So these slides actually showing the different types of symptoms that we recorded, and this is a global data, not a single population data. Here is, we are showing that what is the rate of mortality, and you can see that at the general populations, it is about 10%, but in case of individual Down syndrome cases, because Down syndrome patients do not survive beyond the age 80, so there is no data regarding Down syndrome, but there are some data, and you can see with the more older age Down syndrome people, they have greater chance of mortality in hospitals. And all this data actually showing Down syndrome peoples, these trisomy individuals, they are showing the bulk of the mortality than the euploid individual. So this part of the bar diagram is euploid, representing euploid individuals, and this one is Down syndrome individual. So there are certain other risk factors associated with adverse outcome of the COVID-19 in individual with the Down syndrome. In our survey that came out with these, that means living status, because in our country as well as in Western countries, there is a practice of putting those Down syndrome children into the home, not in the family. So that is another important factor. In case of the home, there is not proper care and maintenance of the social distancing and healthy conditions that lead to the more infections. In different way, we have tested different parameters. That is another important factors that emerge in due course of this survey, that is males are more affected than females. That is a very surprising fact that we found, that in case of the Down syndrome as well as in normal populations, common people populations, the males are more affected. If you go for the statistics, taking statistics even for the sample from Calcutta, you could find that Down syndrome, that the COVID positive patients are usually male for 70% of the cases, and 30% is actually the female. There are some sex-specific difference in the immune response, and that's make this difference. And level of intellectual disability like Alzheimer's or other kind of dementia, that may impose some threats of having infections, thyroid disorder, sugar. These are some of the important. Obstructive sleep apnea. Obstructive sleep apnea means during the course of sleeping there is a problem, nasal problems of taking oxygen. These are some of the important risk factors that make the Down syndrome people more and more susceptible for the COVID infections. So after taking data every month, we meet in virtual meeting, and we try to put this data into the graphical presentations in different languages so that this survey could be spread to the different parts of the world, and common people could understand what we are doing, and they should be cautious. So initially, 12 languages were selected for translating our results in every month bulletin of NIH, and this logo is Trisomy 21 Research Society logo, international logo. So I fought for my own language, that is Bengali, and obviously our national language Hindi. And with those, this one is Spanish and English, and there are Russian and Chinese and Portuguese and so many different languages. And with this, I have succeeded to put Bengali into this queue as well as Hindi in this queue, not shown in the sides, so that with all this, now we have 14 different languages, including Bengali and English, Bengali and Hindi, and all these are in the NIH website. So this is our global task force. She is Stephanie. I am talking about. She is my collaborator at USA. Mara Derson from Spain, and Andres Stridham. He is the leader. He is the leader. He is from London School of Biological Science. He's actually the medical practitioner as well as scientist. This is Monica. Monica also from the London. This is Roger. Roger from USA. He was the initial president of our society, Trisomy 21 Research Society. And this is Louis from France. This is Brian from Portugal. And it's obviously me. There are so many other different members. I'm not giving this all the photographs of this. These are the principal company. We were actually led the entire survey across the world. So the third part of my lecture, and that is the conclusive part, because our research is not only our research in the cold and sophisticated laboratory. Our research obviously carries a social component and social aspects that is important. There are several social issues that we have to handle every time beside our classical research work like PCR, like cloning, like sequencing, gene analysis, statistical modeling. Side by side, at the end of the week, we have to meet, we meet usually with the parents and the Down syndrome children to motivate them, and we provide counseling for them. So that is very important. Unless you translate your research outcome into the applicable figure, you could not make any important impact on the society. So there are some important social aspects, that is family and the parents having Down syndrome baby are stigmatized due to the lack of knowledge. Down syndrome children are not abnormal. So you guys, you have to remember with your fresh and open mind that they are not abnormal. We often called our Down syndrome children from our society, that is maybe their Down syndrome children may be present in our neighbor's home, maybe in our relatives' homes, and we often identify these children are abnormal. আমরা বলি না যে এরা abnormal, এই বাচ্চাটা না একদম normal হয়নি. These are very painful. They are subnormal. That means they are suffering from the developmental delay, so they could not reach out up to the level like all other baby. কিন্তু ওদের সমস্ত feelings থাকে এবং ওদেরকে যদি ট্রেন করা যায়, they could be bring out into the mainstream of the society. Okay, they suffer from the developmental and mental retardations. They are able to carry daily work. They can achieve success. I will show you some of the success story. So this is Paul Socka from USA. He is now awareness leader of the UNESCO. He sought for mentally retarded people across the world. Pablo Pineda, he is a Spanish TV actor, successful actor from the Spain. And Angela Bachelor, she is the former city councilor of the Spain. Okay, Down syndrome, people with the Down syndrome had participated into the politics and came out as a leader. Moria Rossi, she is actress from USA. And Tim Harris, he is the largest KFC outlet owner in California. So there are so many different success stories, and I am happy that now these success stories include some story from India too. So we observe World Down Syndrome Day on 21st March of every year. This year, my team is going to organize one health camp, free health camp for the Down syndrome children, particularly from the marginalized families of our society, because they could not afford the costly health checkup regime every month. So we are going to organize this at University of Calcutta with the financial support from our International Trisomy 21 Research Society. So we need just love and care to bring them back into the mainstream of the society. Sorry. So this is my team at University of Calcutta. It was a great program last year, just before the lockdown initiated. It was in the February 2020, and she is Stephanie, and she is Eleanor. They were there from the USA to make this program successful, and this is my entire research team. And just I told you that often at the weekend we met with Down syndrome family and children to motivate them, to inspire them, to counsel them how to make the life of these Down syndrome children better at our society. So these are my collaborators from India and as well as abroad from the different parts, and have a sweet day. Thank you.
Thank you so much, sir. Thank you for your brilliant presentation. Now it is open for question. If anyone have any question, you can ask Dr. Sujay Ghosh.
Three questions are there already in the drop box.
Okay.
We have three questions. Amritendu, Debanik, and Shivam Chakraborty. They already posted three questions.
Will you please read out the question?
Sure, sir. Sure. Shivam is asking, sir, can you please explain the molecular aging?
Well, Shivani, who is telling that? Shivani.
Shivam.
Aging is something, programmed gradual deteriorations of the biological system. But what happened, usually we estimate age in terms of chronology. That means how much older she is or he is in terms of year. But beyond this estimation, there is another estimation by the nature, how old his molecular system or subcellular system. That means whenever you go counting age with number of the years, that may not actually reflecting the condition that are present in the cell. Molecular aging means the estimations of age in terms of quality of the subcellular molecule and component within the cell. Am I clear?
Thank you, sir. Next one is from Amritendu Ganguly, and he.
But there is some other aspects which were beyond the scope of present lecture. This something very bigger, that is a problem associated with Down syndrome. Down syndrome children suffer from different kinds of physiological and anatomical complications. So one of the anatomical complications is facial dysmorphisms. You know that Down syndrome people have particular very specific type of appearance, and that include malconstruction of the nasal passage in the bone. So these nasal passage with these obstructions create problem during the course of the usual respiratory process, and that goes high during the time of sleep. As you know that we snore usually during sleep, not in the normal conditions when we are in having conditions. So it is happened with the Down syndrome too. Whenever they go for sleep, these nasal passage get obstructed more with the mucus, and immediately they suffer from anorexia and they rise from the sleep. They could not sleep properly. There is a huge disturbance. This is called obstructive sleep apnea.
Okay, thank you, sir. Next, Debanik Dev is asking how contraceptive pills increase the risk factor.
Well, you know that contraceptive pill basically made up of different hormone analogs. And as you know, as a biological science student, that hormone always works in a very minute balance or tighter. If there is an excess hormone, that could create problem. If there is less hormone, that could create problem. whenever a girl is taking contraceptive pill abruptly, he actually allowing huge amount of the estrogen to be there in the body, or estrogen analog to be there in the body, that abruptly rise the estrogen level in the blood, and that estrogen level lead to abnormal molecular signaling in the oocyte and cause non-separation of the chromosome.
Okay, thank you, sir. Next, Pallav is asking, is the rate of molecular aging can be regulated epigenetically?
I don't know. I don't know. That could be done. You can, today we are in the age of different types of microRNA or oligo, synthetic oligo, with which we can stop expression of the different targeted gene. I don't know if there is any way to stop this telomere component so that they could not shorten abruptly or rapidly. I don't know. You people please come to research and you may discover this strategy in your future. I will wait to see your end.
Okay, thank you, sir. Next, Chirantan Maiti is asking, can any person show Down syndrome and Turner syndrome, Klinefelter syndrome together combinedly?
I never heard of. Actually, what happened, wherever there is an error that could affect one chromosome. If, say for example, if theoretically, if you consider that all the chromosomes undergo the non-disjunction junction and create an abnormality, I think that side would not survive. They would die, they would peril within the ovum, within the ovary, without undergoing ovulation. So that oocyte would not come out through the ovulations for normal fertilization.
Okay, thank you, sir. Sir, may I ask one question? Because I'm a bit curious about one thing. Is there any relationship in between Down syndrome, telomeric length and telomerase that could create modality towards cancer?
One thing is that, Krish, that is, Down syndrome people are prone to develop leukemia. Okay. But at the same time, they are resistant to develop any solid cancer, solid tumor. That is very paradoxical. Leukemia is caused by the excess expression of the GATA4, the gene which is located on the chromosome 21, and there is an excess copy of GATA, and that led to the leukemia. But at the same time, due to certain reason, the Down syndrome people cannot develop any solid tumor in their body. So the answer is very, very difficult to say anything right now, but it may possible that telomere and Down syndrome may have some link with the risk of developing cancer, and future work will discover that.
Okay, thank you, sir.
Any other question?
Sir, I have a question.
Please.
Hello.
Yeah.
Sir, is there any relation with the Down syndrome that the parent who become the parent who are older in age can give to a birth to a Down syndrome baby? Is there any relation with the age of the parent?
Yes, I started talking with that. Particularly, age of mother is very important, because if you compare the ovum and sperm, you know that the contribution of ovum is much more in a baby making than a sperm. So maternal age is a big factor. Whenever there is an aging, say a lady decided to take baby at the age of 40. So what happened during that time, the entire protein machinery within her oocyte start deteriorating. That is the natural deteriorations of the protein, and now she will wish to have baby. So what happened, that oocyte have to compromise with all those components that should be there in the optimum number. So there should be defect. Age is the main factor. Paternal age may not be a big factor in making Down syndrome baby, but maternal age obviously is a big factor.
Okay, thank you, sir.
Okay.
The
question. Yes, I think this probably the last question we can take. Shivam Chakraborty is asking, sir, if we remove the GATA gene and insert in embryo to stop cancer, can we do that?
Don't know. There are several mouse models on which different kinds of research, that kind of gene knock-in or knockout research are carried over, but nobody has initiated that type of knockout the GATA copy from the gene and introduce something else. I don't know. Maybe some worker in future may try this line to make the Down syndrome people less susceptible for the cancer or leukemia. I don't know.
Sir, please allow me to ask one thing, that as you have said that more maternal contribution is there for the development of Down syndrome, is there a relationship in between the mitochondrial DNA and the Down syndrome?
Sure. One of my students, he has started some work on the mitochondrial gene polymorphisms. Actually, what happened, mitochondria maintain the respiratory efficiency of the oocyte, and as you know that all the activity, subcellular activity, is somehow energy dependent. So if there is mitochondrial polymorphism, and if that polymorphism leads to compromised respiratory efficiency within the oocyte, that obviously affect the chromosome segregation system, obviously. So one of my students, he started working on the mitochondrial polymorphisms, and we got some very, very interesting results in that regard. But as the publication has not there, so I'm not going to share these results right now. But near future, whenever that will come, sure, in some other occasions, I will meet you and will deliver the results of the mitochondrial gene polymorphisms in the oocyte and its relation with the Down syndrome.
Thank you, sir. Thank you.
Any other question?
Not anything in chat box. Maharaj.
Okay. ঠিক আছে. Then Avijit.
Yes,
Hello.
sir. Yes, Maharaj. So it is a great honor and privilege to propose the vote of thanks on this memorable webinar. First of all, I would like to thank our chief guest who honored this webinar with his valuable talk. We are really fortunate to know the unknown facts of very known Down syndrome. I would also like to thank our beloved principal, Swami Ekochittanandaji Maharaj, our vice principal, Swami Mahapragyanandaji Maharaj, and all the faculty members and participants for making this webinar successful. Thank you everyone. Thank you, Dr. Sujay Ghosh, for this valuable talk.
I would like to add, with our taking permission from our respected HOD sir, AD sir, I would like to add, because we have today, though he didn't come in this Google Meet, but today we have Jeet Maharaj, Ishatma Chaitanya ji. So he is there. He is very instrumental for this. This is also online video streaming is going on, YouTube streaming, and this entire session is recorded and it will be available in YouTube hereafter. So on behalf of the department, We thank our Jit Maharaj also, who was very instrumental and played a very pivotal role. And definitely, I personally, as Principal Maharaj mentioned, I personally look forward to have Sujay Babu in our campus. We already last year we planned for we planned for doing some work, but because of this COVID pandemic, it was not possible. So as the situation restores normalcy, I and all the faculty members and the students, we are eagerly looking forward to have you here physically today. That this webinar is a very humble beginning of this entire procedure. Okay, sir. Thank you. Thank you very much, and thank you, everyone. Namaste. Everyone. Namaste.